Assalamualaikum warahmatullahi wabarakatuh and a very good day. I am Renal from Physiology Department, and in this video I am going to talk about renal physiology. For Renal Physiology 1, we are going to look at glomerular and tubular functions as well as clearance for the assessment of function. For glomerular and tubular functions, we are going to look at glomerular filtration and reabsorption and secretion process that occurs at the tubules. For clearance, we are going to look at renal clearance, as in how to calculate and to use the renal clearance to assess renal function. First, let us revisit the structure of the urinary system. It consists of two kidneys, where urine formation process occur, two ureters that connect kidneys to urinary bladder, one urinary bladder that holds the urine, and urethra that connects the bladder to the body exterior. So kidney, it is a retroperitoneal organ located between T12 to L3. This is a cross-sectional view of a kidney. It consists of three regions, which are cortex, which is the outer region, medulla, which is the middle region, and renal pelvis, which is the innermost region. Ureter, it is a fibromuscular tube. It connects each kidney to urinary bladder. Urinary bladder, it is hollow, sphere-shaped muscular organ in the pelvis, whereas urethra is a small duct that channels urine from the bladder out of the body. Renal form various functions. One of the functions is to regulate water and electrolyte balance. This is done by filtering electrolytes and water from blood, returning some to the blood, and excreting any excess into the urine. Another function of renal is to regulate body fluids osmolarity. This is done by controlling the amount of water excreted in the urine. Another renal function is for maintenance of acid-base balance. This is done by excreting hydrogen ions into the urine and reabsorbing bicarbonate from the urine, or vice versa. Another renal function is to excrete metabolic waste and foreign substances. This is done via secretion process as well as micturition. Renal also has endocrine functions such as to secrete renin and erythropoietin. Renin is important for renin-angiotensin-aldosterone system that mediates the volume of ECF and arterial vasoconstriction, whereas erythropoietin is a hormone that promotes the formation of red blood cells in the bone marrow. So renal has these endocrine functions. Another functions of renal is to regulate vitamin D production. Renal converts inactive vitamin D, either from supplements or from the sun, to active form of vitamin D that can be used by the body. So renal receive about 20% of cardiac output. Blood from aorta enters each kidney via renal artery. Renal artery branches into segmental artery, and segmental artery branches into interlobar artery, which further branches into arcuate artery and cortical radial artery. The smallest arteries subdivide into the first set of arteriole, which is the efferent arteriole. Blood will enter efferent arteriole and enter the glomerular capillary network that will undergo filtration process. And then blood will leave the glomerular capillaries via efferent arteriole, which is the second set of arterioles. From efferent arteriole, the blood will be delivered to a second capillary network, which is the peritubular capillaries, or also called vasa recta. So blood from the vasa recta will flow into cortical radiate vein, arcuate vein, interlobal vein, renal vein, and back to the inferior vena cava. Now we will look at the functional unit of the kidney, which is nephron. There is about 1 to 1.2 millions of nephrons per kidney, and this number will start to decrease after 45 years old. So with this decrease in number of nephrons, there will be age-related decrease in glomerular filtration rate, or GFR. Nephron consists of two main parts, which are renal corpuscle and renal tubule. There are two types of nephrons, which are superficial cortical nephrons and juxtamedullary nephrons. The glomeruli of the superficial cortical nephron is located in the outer cortex. This is cortical layer of the kidney, and this is medullary layer. So the glomeruli of the superficial cortical nephrons is located in the outer cortex, whereas for juxtamedullary nephron, the glomeruli is located near the corticomedullary border. Another difference between superficial cortical nephron and juxtamedullary nephron is the location of the loop of Henle. The superficial cortical nephron have shorter loop of Henle, and the loop of Henle is located in the cortex. Whereas for juxtamedullary nephron, the loop of Henle is long and plunges into the medulla. So renal corpuscle is divided into two portions, which are glomerulus and Bowman's capsule. Glomerulus is a tuft of capillaries connected to an afferent arteriole and efferent arteriole. This is the glomerulus. This is the afferent arteriole, where blood will enter into the capillaries, and from the glomeruli it exits via efferent arteriole. So this is the glomerulus. Bowman's capsule encapsulates this glomerulus. This is Bowman's capsule. It consists of two layers, which are parietal layer, which is the outer layer, and the inner layer, which is visceral layer. This visceral layer consists of podocytes. So filtrates from the glomerulus will enter capsular space. This is the capsular space where filtrates from glomerulus will enter. From this capsular space, the filtrates will enter renal tubule, starting with proximal convoluted tubule. Attached to this renal corpuscle is another structure called juxtaglomerular apparatus. We will look into this structure a little bit later. Apart from renal corpuscle, the nephron also consists of another structure, which is renal tubule. So filtrates from the glomerulus will enter this tubule. Renal tubule consists of proximal tubule, loop of Henle, distal tubule, and collecting duct. Proximal tubule is the first segment of renal tubule. It consists of a coiled segment, often termed as proximal convoluted tubule, followed by a straight segment or proximal straight tubule. Proximal tubule is usually located in the renal cortex. Preceded of proximal straight tubule is the loop of Henle. It is a U-shaped loop. For superficial cortical nephron, the loop of Henle is located in the cortex, whereas for juxtamedullary nephron, the loop of Henle usually plunges into the medulla. The loop of Henle has three functional divisions, which are thin descending limb, thin ascending limb and thick ascending limb. After loop of Henle, there is distal tubule. Distal tubule then empties into the collecting duct. Several collecting ducts converge into a larger papillary duct which empties into minor calyx, where several minor calyces converge to form major calyx whereby urine will pass through before reaching the ureter. Juxtaglomerular apparatus is a microscopic structure situated next to the glomerulus. Juxta is actually a Latin word. It means next to. So juxtaglomerular meaning it is situated next to the glomerulus. So this apparatus located at the junction of the afferent and efferent arteriole and a portion of the distal tubule. So this is afferent arteriole and efferent arteriole, and this is distal tubule. So juxtaglomerular apparatus is located here at the junction of these three structures. The importance of juxtaglomerular apparatus is it regulates the function of each nephron. This apparatus consists of macula densa here, extraglomerular mesangial cells here, and juxtaglomerular cells here. So macula densa is actually the cells that lines the distal tubule. This is distal tubule. Remember, the distal tubule is located here, the one that the loop of Henle empties and it enters distal tubule. So the macula densa is actually the cells that lines the distal tubule. It connects the distal tubule to the glomerular. So the importance of macula densa is it acts as sodium sensors, meaning that it is important in tubuloglomerular feedback. It will sense either increase or decrease in sodium chloride contents in the blood or in the lumen, so it will adjust accordingly. So the function of the macula densa, it influences the afferent arteriole either to constrict or dilate. If there is increase in sodium chloride, this will cause afferent arteriole to constrict. This constriction of afferent arteriole will cause decrease in GFR. Whereas if there is decrease in sodium chloride level, macula densa will cause afferent arteriole to dilate. This dilation will cause increase in GFR. So this is how the macula densa acts as sodium sensors. So another cells is extraglomerular mesangial cells here. So one of the function of these cells is to secrete mesangial matrix that help to support the capillary tuft. Or in other words, it helps to stabilize the glomerular endothelial function. And another cell is juxtaglomerular cells. Here, this is actually the cells that line the afferent arteriole. They are specialized smooth muscle cells of the afferent arteriole. So these juxtaglomerular cells secretes renin, which is important in the renin-angiotensin-aldosterone system. There are three basic processes of urine formation. This figure illustrates this process. This is the glomerular, and this is the tubule. This is peritubular capillary. The first process is glomerular filtration, where blood in the glomerular capillaries is filtered through the filtration membrane. So the filtrates will enter the tubule starting from proximal tubule, whereas the unfiltered substances will flow into the efferent arteriole and to the peritubular capillary. Second process is reabsorption. For reabsorption, solutes such as sodium, chloride, bicarbonate, glucose, and urea, and also water, are removed from the tubular lumen and transported back into the peritubular capillary. So this reabsorption process occurs throughout the renal tubule. Depends on several factors such as the type of substances, availability of transporter, and also availability of hormone. The third process is secretion process, where the unwanted substances from the peritubular capillaries will be secreted into the tubular lumen. Similar with reabsorption process, the secretion mechanism also involves transporters. The next process is excretion process, which is the elimination of urine from the collecting duct to the ureter via renal pelvis. First, we are going to look at the first process in urine formation, which is glomerular filtration. It is a process by which the blood that passes through glomerular capillaries is filtered through the filtration membrane or filtration barrier. So the filtration barrier consists of 3 layers, which are capillary endothelium, capillary basement membrane, and food processes of podocyte of the glomerular capsule. So blood from efferent arteriole that flow into the glomerular capillaries will be filtered through these 3 layers of filtration barrier and enter the Bowman's capsule, capsular space of Bowman's capsule, whereas the substances that cannot be filtered will flow back into the efferent arteriole. There are 2 important factors that determines the type of substances filtration across the filtration barrier. One of the factors is molecular size. Smaller molecules will be filtered more easily compared to bigger molecules. For example, small solutes like sodium, potassium, chloride, or bicarbonate are freely filtered across the filtration barrier as opposed to bigger molecules like albumin. The molecular size cutoff of the filtration barrier is about 70,000 kilodalton, meaning if the molecule has a molecular weight lower than 70,000 kilodalton, it may be able to be filtered across the filtration barrier. Albumin, for example, has a molecular weight of 69,000 kilodalton. So, by right, albumin is able to be filtered and may appear in the urine. However, this does not normally occur because another factor that determines the ability of a solute to penetrate is the ionic charge. Cations, which are positively charged molecules, has higher filterability or easier to be filtered as opposed to anions. This is because podocytes are polyanions, which is negatively charged. So, in the case of albumin is an anion, which is negatively charged. So, although its molecular weight is smaller than the filtration slit of the podocytes, because it is negatively charged, it cannot pass through the podocytes. So, these 2 factors, molecular size and ionic charge, determines whether a substance can pass through the filtration barrier or not. The glomerular filtrate, which is the filtrate in the Bowman's capsule, contains most inorganic ions and low molecular weight organic solutes. The filtrate normally does not contain any cells, and it has almost similar concentrations to the plasma. The flow rate of filtered fluid through the filtration barrier is termed as glomerular filtration rate or GFR. Estimated GFR is the best test to measure the level of kidney function and to determine the stage of kidney disease. GFR is the total volume of fluid filtered by all glomeruli in both kidneys per 1 minute. It approximately 125 milliliter per minute or 180 liter per day in an adult, but this depends on the body surface area. Smaller person may have lower GFR than a bigger person. Similar to the systemic capillaries, the pressures that drive fluid movement across the glomerular capillary wall are the stalling pressures or stalling forces. Stalling pressures of the systemic capillaries consist of 4 pressures, which are 2 hydrostatic pressures, 1 in capillary blood and 1 in interstitial fluid, and 2 oncotic pressures, 1 in capillary blood and 1 in interstitial fluid. However, when applying these pressures to glomerular capillaries, there is one small modification. The oncotic pressure of Bowman's space, which is analogous to interstitial fluid, is considered to be 0 because filtration of protein is negligible. Remember that protein is not filtered. So, the oncotic pressure of Bowman's space is considered to be 0. PGC is the hydrostatic pressure in the glomerular capillary. PBC or PBS is hydrostatic pressure in the Bowman space, and PIGC, the oncotic pressure in the glomerular. So glomerular filtration rate depends on the balance between the hydrostatic pressure gradient and the oncotic pressure gradient across the capillary. The balance of these forces allow calculation of the net driving pressure for filtration. Stalling equation, GFR is equal to Kf, which is filtration coefficient, times net filtration pressure. Filtration coefficient, Kf, is the water permeability or hydraulic conductance of the glomerular capillary wall. The two factors that contribute to Kf are the water permeability of the glomerular capillary wall and the total surface area of the glomerular capillary wall. It is approximately 12.5 milliliter per minute per millimeter mercury. Net filtration pressure, it is the stalling pressures across capillary wall. The net filtration pressure is determined by the 3 pressures: glomerular hydrostatic pressure, glomerular oncotic pressure, and Bowman's capsule hydrostatic pressure. So glomerular hydrostatic pressure is exerted by the blood in the glomerular capillaries. This pressure favors filtration. Notice that the arrow from the glomerular is directed into the Bowman's capsule, meaning that it favors filtration. Another factor is glomerular oncotic pressure. This is exerted by plasma proteins in the glomeruli. And this pressure opposes filtration, meaning if the oncotic pressure is high, GFR will be low. And the third factor is Bowman's capsule hydrostatic pressure. This pressure also opposes filtration. So to calculate the net filtration pressure, glomerular hydrostatic pressure minus Bowman's capsule pressure minus glomerular oncotic pressure. So in this example, 60 mm mercury of glomerular hydrostatic pressure minus 18 mm mercury minus 32 mm mercury. So the sum of net filtration pressure is 10 mm mercury. So in summary, GFR is equal to Kf, filtration coefficient, times the net filtration pressure, which are glomerular hydrostatic pressure minus Bowman's capsule hydrostatic pressure minus glomerular oncotic pressure. So this is how GFR is calculated. There are many factors that may affect GFR. One of the factors is constriction and dilation of afferent and efferent arteriole. So this is an example of the effect of constriction and dilation. So in A, there is constriction of afferent arteriole. When there is constriction, resistance will increase. So this will cause hydrostatic pressure in the glomeruli to decrease, and this will cause decrease in GFR. Remember that the GFR is proportional to the hydrostatic pressure in the glomerular. So decrease in hydrostatic pressure of the glomerular will cause GFR to decrease. In B, the constriction is on the efferent arteriole. This will cause glomerular hydrostatic pressure to increase, which will cause GFR to also increase. The effect of dilation is the opposite of constriction. Dilation of efferent arteriole will decrease GFR, whereas dilation of afferent arteriole will increase GFR. Apart from constriction and dilation of the arteriole, there are other factors that may affect GFR, such as changes in renal blood flow, changes in systemic blood pressure, urethral obstruction. In the case of urethral obstruction, for example, presence of urethral stone. The presence of stone will cause urine cannot flow through the ureter to the bladder. This will cause urine to stay in the kidney. That will cause the Bowman's capsule hydrostatic pressure to increase. The increase in Bowman's capsule hydrostatic pressure will cause decrease in GFR. Another factor, for example, is edema of kidney. Edema of kidney will cause increase in hydrostatic pressure in the Bowman's capsule as well. So this will cause decrease in GFR. And other factors such as changes in KF, which include glomerular capillary permeability and effective filtration surface area. So how do we measure GFR clinically? We do know that it cannot be measured directly. So GFR is measured by the clearance of a glomerular marker. A glomerular marker should have all the following characteristics, which are freely filtered across glomerular capillaries, and it cannot be reabsorbed or secreted by the renal tubule. Another characteristic is when the glomerular marker is infused into the body, it should not alter the GFR. So GFR can be measured by calculating the renal clearance, which is the ratio between the renal excretion rate of a substance to its concentration in the plasma. We will look at how to measure this renal clearance later. Inulin is the ideal indicator or gold standard in GFR determination. This is because inulin fulfills all the criteria. It is freely filtered, not reabsorbed, not secreted, and not metabolized. Plus, it is not toxic to the body. However, it is not an endogenous substance, meaning that it is not produced in the body, so it needs to be infused intravenously. So in clinical practice, inulin is not usually used, but instead creatinine is used. Creatinine is produced naturally by the body, and also it is freely filtered by the glomerulus. So GFR is equal to urine concentration of substance X. Urine concentration of X. X can be anything that we want to measure, be it insulin, creatinine, or any other substance that you want to measure the clearance. So urine concentration of X times urine flow divided by plasma concentration of that substance. So to estimate GFR using creatinine clearance, we need to get three measurements, which are urine creatinine concentration, 24-hour urine volume, and plasma creatinine concentration. So for example, the creatinine in the urine is 1.25 mg per milliliter, creatinine in the plasma is 0.01 mg per milliliter, and flow rate of urine flow is 1 milliliter per minute. So GFR is 125 milliliter per minute. So for example, if there is decrease in GFR, for example in renal failure, serum or plasma creatinine is increased. This is indicative of renal failure because this creatinine is not filtered adequately. So this will cause plasma creatinine to increase, and it is indicative of renal failure. Other than creatinine, blood urea nitrogen (BUN) may also be used to estimate GFR. So we have covered glomerular filtration. Now we will look into the next process in urine formation, which are reabsorption and secretion process. The tubular reabsorption occurs via two basic transport mechanisms, which are paracellular pathway, which is between the cells, and transcellular pathway, which is through the cells. For paracellular pathway, the filtrates or substances in the lumen pass through the epithelial layer of the tubule in between the tubular cells and into the peritubular capillary. Whereas for transcellular pathway, the way the substances reabsorb from the lumen to the peritubular capillary is through the tubular cells. This paracellular in between the cells and transcellular pathway through the cells. So these are the pathways that substances can get through from lumen into the tubular capillary. The mechanisms by which this transportation occur depends on the type of substances, transporters, and the need of energy. The transport mechanisms that does not need the use of energy are diffusion, which can be further categorized into simple diffusion and facilitated diffusion. So both simple and facilitated diffusion are passive processes that does not require energy. Solutes or molecules move from higher concentration to the lower concentration. In the reabsorption process in the tubules, if the concentration of the substance is higher in the tubule, the substance will diffuse from the lumen into the tubular capillary, either via paracellular or transcellular pathway. For facilitated diffusion, it depends on transporters or carrier proteins. These carrier proteins are located on the tubular cells. Whereas for water, it is transported via osmosis, either paracellularly or transcellularly. Tubular reabsorption may also occur via active process, which are primary and secondary active process. In active transport, the concentration of the substance may be higher in the tubular cells or capillary than in the lumen. So the concentration of the substance will be lower in the lumen compared to the tubular cells and peritubular capillary. So this reabsorption process will require energy. In primary active transport, the energy comes from ATP. Whereas in secondary active transport, the energy comes from the primary active transport. There are two types of secondary active transport, which are co-transport and counter-transport. For co-transport, the two molecules are translocated to the same direction, whereas for counter-transport, the two molecules are translocated on the opposite direction. We need to understand that each of the renal tubule have their own unique characteristics. For example, some part may have certain type of transporters, while others do not. So different tubules may absorb different type of substances or solutes. So let us look into this reabsorption process, tubule by tubule. Proximal tubule reabsorb about 65% of the filtered water, sodium, chloride, and potassium. It also absorbs about 90% of filtered bicarbonate, almost all glucose and amino acids. This means that when the filtrate leaves the proximal tubule, it usually does not contain any glucose or amino acids. There are presence of extensive brush border, which increases the surface area for absorption. There are also some secretion process going on in the proximal tubule. Secretion means that the substance moves from peritubular capillary into the lumen. Proximal tubule secretes excess hydrogen ions, organic acids, and bases. Reabsorption process in the proximal tubule occur mainly via active process, either primary or secondary active transport. The reabsorption process is quite different in the early proximal tubule and late proximal tubule. So this is the cell of the proximal tubule. This is the lumen, meaning that this is the luminal membrane of the cells, and this is the blood in the peritubular capillary, meaning that this is the basolateral membrane. So in the early proximal tubule, sodium is reabsorbed from the lumen into the cells via co-transport, such as sodium glucose co-transporter. This is also called SGLT, which stands for sodium dependent glucose transporter. There is also other co-transporters such as sodium amino acid co-transporter, sodium phosphate co-transporter, sodium lactate, sodium citrate co-transporter. So sodium is then extruded from the cell into the blood by the sodium-potassium ATPase located at the basolateral membrane. Whereas glucose and other solutes that were co-transported together earlier will be extruded via facilitated diffusion. There is one counter-transport in the luminal membrane of the early proximal tubule, which are sodium-hydrogen exchanger. Sodium is reabsorbed into the cell in exchange for hydrogen. This will cause secretion of bicarbonate from the cell into the blood. So this is in the early proximal tubule. In the late proximal tubule, the substances that are majorly reabsorbed are sodium chloride. Because when the filtrates reach the late proximal tubule, all the filtered glucose and amino acids have been reabsorbed into the blood. So when it reached the late proximal tubule, it does not contain any more glucose and amino acids. But on the other hand, it has a high level of sodium chloride. So chloride is reabsorbed via paracellular pathway, via diffusion, or transcellular pathway via chloride format anion exchanger. For potassium, the reabsorption of potassium at the proximal tubule is mostly via passive diffusion. This is the diffusion process of potassium. It can be either from the cells into the lumen or into the blood. There are also sodium-potassium ATPase pump that pump sodium out of the cells and potassium into the cells. But the higher level of potassium in the cells will cause potassium to diffuse out of the cells, either to the lumen or into the interstitium or blood. For bicarbonate, about 90% of bicarbonate is reabsorbed in the proximal tubule, whereas the other 10% is reabsorbed in the distal tubule and collecting duct. Bicarbonate is reabsorbed via passive diffusion. However, bicarbonate ion cannot cross the luminal membrane because it is negatively charged. Therefore, the bicarbonate will combine with hydrogen ion to form carbonic acid. The carbonic acid will be dissociated into carbon dioxide and water by carbonic anhydrase. Carbon dioxide is lipid soluble, so it can easily enter the proximal tubular cells. In the tubular cells, the opposite process occurs. Carbonic anhydrase will combine carbon dioxide with water to form carbonic acid. Carbonic acid will dissociate to form hydrogen ion and bicarbonate ion. On the basolateral membrane, there are sodium bicarbonate co-transporter and bicarbonate chloride counter-transporter. So bicarbonate can be reabsorbed into the blood via these transporters. Reabsorption of water. Proximal tubule is highly permeable to water. meaning that it does not need any hormones such as ADH to alter its permeability to water because it is already permeable to water. So water will be reabsorbed into the capillary blood from the lumen into the capillary blood because of higher osmolarity or higher osmotic pressure in the capillary. This higher osmolarity of blood is contributed to the solute reabsorption that occurred earlier. Remember that at early proximal tubule, sodium reabsorption occurs. So, the positive charge in the blood will attract negatively charged ions such as phosphate and bicarbonate to also enter the blood. So, this will cause increase in osmotic pressure in the blood. Hence, water will be reabsorbed from the lumen into the blood via osmosis. This process is not regulated by ADH. Those are the reabsorption processes that occur in the proximal tubule. At loop of Henle, different reabsorption processes occur. The loop of Henle consists of 3 limbs: thin descending limb, thin ascending limb, and thick ascending limb. Thin descending limb is highly permeable to water, so it allows water reabsorption via aquaporin 1 water channels. About 20% of filtered water will be reabsorbed at the thin descending limb. Thin descending limb is also moderately permeable to solutes including urea and sodium. However, it does not reabsorb significant amount of solutes because it does not have any transporter or carrier proteins, so the solutes will be reabsorbed via simple diffusion. The permeability of the thin descending limb to urea is important for urea recycling process. On the other hand, the thin ascending limb, unlike the thin descending limb, it is not permeable to water, meaning water is not reabsorbed at this limb. This impermeability of the ascending limb to water is important for countercurrent multiplication process. This limb is also permeable to urea. So this permeability to urea is also important for urea recycling process. We will look into these two processes, countercurrent multiplication and urea recycling process, in another video. Similar to the thin descending limb, the thin ascending limb does not have transporter, so it does not reabsorb significant amount of solutes. Its reabsorptive capacity is much lower than the thick ascending limb. Similar to the thin ascending limb, the thick ascending limb is also impermeable to water. However, the thick ascending limb reabsorbs a significant amount of solutes because it has many types of transporters. One of the transporters is sodium potassium 2-chloride cotransporter, or NKCC2. NKCC1 is located throughout the body, but NKCC2 is located specifically in the kidney. This NKCC2 allows reabsorption of sodium, potassium, and chloride into the tubular cells. Higher potassium in the cell will cause backleak of potassium back into the lumen, causing the tubular fluid to be positively charged. This positive charge of tubular lumen will force or attract cations such as magnesium, calcium to diffuse into the interstitial fluid or into the blood via paracellular diffusion. Early distal tubule, it has the same reabsorptive characteristic of the thick ascending limb, meaning it is also not permeable to water, so it does not absorb water, and it absorbs significant amount of solutes such as sodium, potassium, chloride, calcium, and magnesium, using cotransporters such as sodium chloride cotransporter. Both late distal tubule and collecting duct have similar functional characteristics. There are two cell types on the tubular lining, which are principal cells and intercalated cell. These two cell types reabsorb and secrete different solutes. Principal cells reabsorb sodium and secrete potassium, whereas intercalated cells reabsorb potassium and bicarbonate and secrete hydrogen. At the basolateral membrane of principal cell, there is sodium-potassium ATPase pump that pumps sodium out of the cells and potassium into the cells. This will cause sodium concentration inside the cells to be lower than the tubular lumen. This will cause sodium to diffuse into the cell via epithelial sodium channel, ENaC. This process is governed by aldosterone. Presence of aldosterone will stimulate reabsorption of potassium by the principal cells. Intercalated cells, the basolateral membrane contains bicarbonate chloride countertransporter, so the function of this intercalated cell is to reabsorb bicarbonate. On the luminal membrane, there is hydrogen ATPase pump. It actively secreted hydrogen from the cell into the tubular lumen. So aldosterone plays an important role in the reabsorption and secretion process in the late distal tubule and collecting duct. There are two important functions of aldosterone in these two tubules. In the principal cells, it increases the activity of sodium-potassium ATPase, which will cause sodium to be reabsorbed and potassium to be secreted. In the intercalated cells, aldosterone increases the hydrogen ATPase activity, which will cause secretion of hydrogen into the tubular lumen. With regards to water reabsorption of these two tubules, both late distal tubule and collecting duct are not permeable to water. However, in the presence of ADH or vasopressin, the permeability of this tubule to water will increase. ADH will bind to vasopressin receptor, and this will activate CAMP, causing insertion of aquaporine or water channels into the apical membrane. This will allow water to be reabsorbed into the blood via osmosis. So, even though they are not permeable to water, with the presence of ADH, they become permeable to water. So water will be reabsorbed by osmosis. This figure illustrates the importance of ADH in urine production. In the first situation, which is a hypothetical situation that our body does not produce ADH, or in the case of diabetes insipidus, the tubules that are permeable to water without ADH are proximal tubule and thin descending tubule only. So water will be reabsorbed here, only here. At the thick ascending limb, a significant amount of solids are reabsorbed. So this will cause the luminal fluid that pass through the distal tubule and collecting duct to be hypoosmolar or diluted. So urine that will produce will not only be diluted but also in large quantity. In the presence of ADH, ADH will increase permeability of distal tubule and collecting duct to water. Hence, water will be reabsorbed. This will cause production of small volume of concentrated urine. So that was reabsorption. A little bit on tubular secretion. It is the reverse process of tubular reabsorption, meaning the substances are transported from blood, which is from the peritubular capillaries, into the tubular cells and into the tubular lumen. The substances that is secreted include potassium, hydrogen, ammonium, creatinine, urea, and some hormones and drugs, for example, penicillin. The secretion is important for eliminating unwanted or undesirable substances, eliminating substances that were passively reabsorbed but need to be removed, for example, urea and uric acid. It is also important to control blood pH, and to eliminate excess potassium in the body. This figure summarizes the reabsorption and secretion process that occur in the tubules. Reabsorption and secretion process mainly occur in the proximal tubule and distal tubule. Sodium chloride are reabsorbed at all parts of the tubules except for the thin descending limb. But the thin descending limb is permeable to water even without ADH, which allow reabsorption of water. Whereas for distal tubule and collecting ducts, they are not permeable to water. They only become permeable to water with the presence of ADH. So water will be reabsorbed here, which allow the production of small volume but concentrated urine. In summary, urinary excretion or amount excreted E is equal to amount filtered F by the glomeruli, this is glomerular filtration, minus amount reabsorbed R, which is reabsorption from lumen to the blood, plus amount secreted S, which equal to urinary excretion. Amount excreted equal to amount filtered minus amount reabsorbed plus amount secreted. So there you have it, renal physiology lecture that covers renal clearance and glomerular and tubular functions. If you have any questions, do email me. Thank you.